She will tell us about the next generation of fish in a mixed methods, I would say.
So, hi everyone, my name is Diana, I'm from Lisbon, Portugal, and I work at IST and IBB, the Institute for Bioengineering and Biosciences. And yeah, today I bring you our work regarding fish fillets and specifically on bioprinting combined with electrospinning and electrical stimulation. So at IST, we actually are growing a quite interesting environment regarding cultivated seafood. We actually started in 2021 with this project funded by GFI, the Algae to Fish project. But we actually have now a large community working on this and PhD students as well. So I'm very proud of the foundation of Cell Agri Portugal and the University of Lisbon Alt Protein Project. And we actually held our first conference last month in Braga. And yeah, this is very exciting. So yeah, today we are here about the cultivated meat, and it actually has very good benefits. I would like to highlight food safety, animal welfare, and accessibility to food. And we all know how to do this. But today I will focus on tissue organization and specifically to distinguish non-organized structures from organized structures, and for example distinguish hamburgers from steaks or fish fingers from fish fillets. And as a tissue engineer, I would focus, of course, on scaffolding, and I will bring you our scaffolding approach to this. So our aim is to combine bioprinting with electrospinning and also electrical stimulation assays. The project actually has a huge part before that. So we actually work on embryonic seed-base expansion, differentiation and characterization, the development of bioinks and electrofibers, electroconductive fibers that I will talk a bit today, and the combination of them and electrical assays. So, and why should we do all of this? Basically, this is the native fish. This is more or less the fillet. We have this myomere here. This is a repetitive structure that happens in the fillet, and it's basically muscle with some intramuscular fat as well, and it's separated by a myoseptum that is actually connective tissue, and this connective tissue it's made of collagen. So we aim to do the bioprinting to mimic the muscle and the fat, and then electrospinning to mimic the fibers, and the electrical stimulation because electrical impulses are actually present in muscle development, and we want to test this during myogenesis. So starting on that, and why should we do that? Basically, there are some literature correlating electrical stimulation impacts on muscle, like for example, cell alignment, cell width, cell length, number of nuclei per cell, with the tenderness and the texture of the meat. And to do that, we actually use the Atlantic macroskeletal muscle cell line that was developed on David Kaplan lab by Michael, and we are following the same protocols basically, but in the last 4 days of differentiation, we actually are comparing AC stimulation with pulse stimulation here, and so basically this is the cells growing on our lab at day 0, and then with the electrical stimulation compared to no electrical stimulation. Here are some of the pictures that we are getting. So then what we do is to try to assess the features that I mentioned before, analyzing these pictures, and we actually saw some difference regarding the number of nuclei per cell, specifically during AC stimulation compared to no stimulation, and then regarding the length and the width in both cases. So move. Oh, this is not working. Moving on to electrical spun fibers. This is basically how we do that. How we do this is our setup. We have this syringe full of our polymer material, and then the solution goes through this tube until a needle, and basically what happens is the solvent evaporates, and there's this electrical field, actually very intense, about 20 to 30 kilovolts, that allows this jet to form until our collector. Very simple way to explain this, but basically we have two different collectors, a circular one that allow us to collect random fibers, and then a parallel one that allow us to collect aligned fibers. And here are some of our examples. We are doing this with zein, that is the protein from corn, and we actually are using non-toxic solvents as acetic acid and ethanol, and we screen for a lot of concentrations of both solvent and polymer, and this is some of our nanofibers under SAM, and we actually were able to calculate the diameters that we were obtaining, and we seeded some embryonic sea bass stem cells on these fibers. They actually adhered very well with no coatings, so this was quite promising, but we actually needed to enhance a bit the mechanical properties of the fiber. We end up to add a bit of gelatin and then microalgae for the organoleptical properties as the smell. So we calculated the diameters again. You can see that they are a bit larger, and actually the mechanical properties were quite good, and we aim to replace this gelatin by microbial one in the future. So going on to the 3D bioprinting, it all starts with bioink development. What we do is basically mix the polymers, media, and cells. We work with kappa carrageen. Basically, it's present in all our bioinks. We then follow for a rheological assessment to understand what's going on during the printing process, printability evaluation, and bioprinting assessment, where we culture these structures during 7, 10, 15 days. It depends. And then we do the live dead assessment for the viability. So we wanted to construct bioinks for fat and bioinks for muscle. And for that, in the fat structures, we actually end up to add plant-based oils and microalgae extracts that smelled like fish. And those are our structures. We did the Young's modulus assessment as well, and we compared to salmon fat. And we actually did viability assays with the like cell line that I mentioned before. And regarding the muscle, what we did was to add plant-based proteins and some pigments, and actually to tune the visual appearance of these scaffolds. And we actually were seeing some colors a bit more like salmon or tuna or even whitefish. So with the bioinks, we are finally ready for the bioprinting. What we do is basically design our own cod models, and that allow us for the combination of muscle and fat. And basically, this is converted in an STL file. We do the slicing. This generates a G code that our homemade bioprinter reads, and then we construct our prototypes. This was our first printing. We then needed to see if all the bioinks were actually compatible with this, and we tried for different muscle bioinks and fat bioinks. So there's our first sashimi, but actually our aim was not the sashimi, was the whole cut fish fillet. So we needed to design something a bit more complex, and we did this CAD design a bit more with curvatures, and it was very interesting. We saw that it was better to print them in vertically, and then in the end of the printing we turn it over like it's here in the plate, and it actually allowed us to combine the muscle and the fat as well here. So this was the prototype. It took like 8 hours to print, but you can see it more closely. Actually, it's very nice to see the fat within the muscle here. So yeah, we did that. So it remains a combination of bioprinting and electrospinning that is basically this. But in here, what we did was to combine actually the muscle bioink and the fat bioink in the same part of the prototype to mimic like the intramuscular fat part, and then in the white part we did those electrospun fibers to mimic the myoseptum that I mentioned before, And so this is very preliminary, it's under development, but you can see at your left, it's the prototype only with the muscle, without any fiber, and then with the fiber at the center and the right, and we actually were very happy. And I brought here a video to show us, because actually it turned the prototype much more real, and yeah. I think it's very nice. And then we did a SAM to actually saw what's happening in the interface of this prototype, and we actually have the fiber region and the bioink region. So in the future, we are actually doing a lot of stuff, but we want to assess how electrical stimulation does, for example, for mobility. And we have this crazy idea of a fish fillet swimming. And we have ideas for novel bioreactors regarding specifically, for example, the electrical stimulation during the culture in a reactor, coaxial electrospinning, a sensorial analysis that will be performed both in Portugal, but as well in a university at Bra, gastronomic science in Italy. And we actually had projects starting in 3D cryo bioprinting that will enable to construct some channels within the structures. And that's it. I really need to thank to our team and to Professor Frederico Ferreira to support this project since the beginning. Of course, Carlos, Paola, Dr. Joaquim Cabral, yeah, all my colleagues and the master students, and the funding as well from GFI. And yeah, thank you very much.
Thank you very much. It all looks very beautiful. Are there any questions?
Thanks for the great talk. So the scaffold that you showed, they also contain cells, or they were only scaffolds? And if they did contain cell, did you see if the cell actually grow inside the scaffolds?
Yeah, we have the cell assessment for all the bioinks, as you saw, like in the first slide. I know, but actually the big prototype, we don't have cells on them. We did the cell assessments, but we need actually to do scales, like very much more tiny scales to do that, because the quantity of cells that you would need for that prototype, it's huge, because we use around 10 to 15 million cells per ml. So we don't have the scale to do that right now. So when we do the bioprinting, with the cells, we do like scaled versions.
So the final vision is to include the cells regardless of having enough number of cells. Do you think that such a big construct can be 3D printed with cells and let the cells grow there? This is the final vision? Or
yeah.
Okay.
Yeah, and that's why we started now the works on 3D cryo bioprinting, because actually it will allow us to construct these channels within the scaffold to allow irrigation and the cells to grow. But actually all bioinks were characterized for cell viability and for both cell lines.
Great, thanks.
Question over there, yeah?
Thank you for your presentation. I'm very curious about your electrical stimulation. So, did you make the device yourself, or?
Yeah.
Can you show me the picture? I saw just a quick.
I did not show the device, okay. But what we used for those stimulations, it's basically a 6-well plate with electrodes on the cap for each well. So, and the cells are seated, and actually the electrode goes within like the culture media, and we then define the parameters for electrical stimulation. And for the scaffold, our aim is actually to construct this reactor where the electrode goes within the structure.
So the whole scaffold with the cells were put under the stimulation, right? Or just the cells were under the stimulation?
The picture that I show regarding electrical stimulation is just the cells.
Just the cells?
Yeah.
And then so you got the cells and then put the cells into the scaffold or doing something else, right?
Sorry?
I mean, like, so what's the main point for doing the stimulation for the cells? Just to get to better differentiation?
Yeah, the main point of electrical stimulation is actually to improve the quality of the muscle fibers that we are getting, and specifically regarding alignment, because we actually think that it can better the texture of the product. And we actually are doing electrical stimulation with just the cells, electrical stimulation within the scaffold, and specifically in the fibers. So we are trying to, our main objective is to have like these aligned fibers where we can see the fish cells and do the electrical stimulation as well to improve the alignment.
Yeah. Were you considering about the whether the whole field was uniform or not? I mean the electrical field.
Okay. We are not actually modeling the electrical field and what's happening there yet. We are going to do that during the fabrication of the reactor.
Okay, yeah, thank you.
Yeah, no problem.
Thanks again, Diana.